there's two ways to get to an answer. You can get the right answer, or you can eliminate all of the wrong answers, right? And so which one you decide to lean on more is dependent on what you know.
Pooja, welcome to the Mcat podcast. Hello, hello.
Hello. Thank you for having me.
I'm excited to chat with you. I think we've chatted once before for something else, but now diving into the Mcat Diagnostic from Blueprint. When it comes to the Mcat for you, what is your top strategy for helping students realize mistakes they're making with the Mcat?
Yeah, I think it's a really good question, especially to ask yourself in the very beginning. And so my biggest piece of advice for students is as soon as you get started, don't just assume you have to go through everything with the same amount of dedication to each and every single topic, right? You have your own strengths and weaknesses from your own science background, from your classes, etc. And so you want to make sure that you're dedicating time proportional to how much you actually need to know the information, as well as how weak you are in that material, right? So like physics, a lot of people are really weak in it, but you don't actually have to know a ton of physics compared to psych-soc, for example, which is probably, in my opinion, one of the most neglected topics because everyone's like, oh, it's easy. But like, that's a quarter of your test, guys. Like, that's a lot of material to just shave off to the end. You know what I mean?
Yeah.
So I think spending time proportionally would be my biggest advice.
Okay. So proportionally to what the test is, right? Not proportionally to maybe your weaknesses or your strengths or whatever.
Right. Yeah.
Got it. All right. Well, with that, let's go ahead and continue our fun progression of the Mcat Diagnostic from Blueprint, starting with passage 3. Why don't you start there?
All right. So passage 3, I'll be reading the first paragraph, right?
Yep.
Okay. Beta-lactam antibiotics like penicillin are derived from fungi, which use the molecules to defend against environmentally borne diseases. Once discovered, these antimicrobial agents were quickly employed by physicians to treat pathogens which invade humans. These molecules are typically formed via intramolecular attack with an amino acid and secondary reactions between amino acids. The lactam designation refers to the cyclic amide ring found in the molecule. Penicillin-like drugs are analogs of amino acid residues located on bacterial cell walls. The structural similarity between beta-lactam antibiotics and these residues facilitates their binding to the active sites in the machinery required to build the peptidoglycan wall.
All right. So when you're reading a paragraph like this, what do you typically like to do? How do you teach students to work through these?
Yeah, absolutely. I think one of the biggest things to point out here is in a lot of Chem Phys passages, they'll throw in a lot of biochem relevant passages. So it's going to be really tempting to spend a lot of attention to the antimicrobial agents or the potential immune function. But because we're in the Chem Phys section, you want to make sure you're looking at this passage through a Chem Phys lens. So you want to highlight the things that are MCAT relevant for the Chem Phys section, right? So paying attention to the specific terms that are being introduced, like beta-lactam antibiotics, is helpful, but also highlighting those terms like intramolecular attack, structural similarity, and amino acids is going to be really important there.
Yeah. So you get through this first one. We are left with this little figure one. Do you pay any attention to figures as you're going through, or you just wait to see if there's a question that asks about a figure?
I like to recognize or sort of like mental map or flag what that figure is. So I would see the figure, I look at that little caption that they have and be like, all right, cool, this is penicillin. And then move on from there. I think spending a lot of time trying to dive into what's particularly present isn't a good use of your time because, first of all, you don't even know if the MCAT's going to ask about it. And second of all, you don't know what specific components of a figure is going to be asked about. So it's not really worth your time to do that in your initial read. These beta-lactam molecules are particularly reactive due in part to significant ring strain. Nearby nucleophiles can be acylated by the ring, allowing the ring to open. The antimicrobial efficacy of penicillin lies in its ability to acylate serine residues on enzymes that bacteria require for normal cell wall production. During the acylation of serine, the amide on penicillin is converted to an ester. This causes binding and locking of the molecules, which prevents serine from activating the cell wall, synthesizing enzymes. An intact cell wall is necessary for bacteria to stay alive.
All right, and then we're led to another figure there. Anything big in this second paragraph that you're like, uh-oh, let's highlight that?
Well, for the second paragraph, again, we get some more information about the general molecule in present, right? So there's a lot of mumbo jumbo.
Yep.
But we want to stay rooted in those Mcat hot topics. So I would probably pay attention to reactive due to significant ring strain, because understanding the relationship of reactivity and how general compounds work in terms of reactions is Mcat relevant. I would also pay a lot of attention to the idea that there's that acylation of serine residues and that we're converting an amide to an ester. So highlighting something to encapsulate that would be really important.
Okay.
I think another thing that's worth mentioning here is that even if you're not 100% sure what an acylation reaction is, that's okay. There's going to be a lot of material that shows up on passages that you're not going to be intimately familiar with, and that's half of the battle. I feel like, I don't know, Ryan, if you agree with this, but I feel like the Mcat is half actually testing the Mcat and then half psychological. Can you be okay with not knowing half of the material that's thrown at you?
A 1,000%. I think there's so much fear around, oh my gosh, I don't know that. Oh my gosh, I don't know this. And it just takes up so much bandwidth in your brain that you're just ignoring everything you're reading because you're like 3 sentences in the past that you didn't know one single word. And this comes up a ton in the CARS section for ESL students. They get so caught up, students in general, and especially ESL students, with a definition they don't know from a word, or an idiom maybe that they don't know in their natural language, their natural tongue, that they're just like, I don't know what that means. Skip it, right? Realize that you don't know it and skip it. And you may not need to know it at all, or context in the question or answers may help lead you to the answer that you need to get. So there's so much focus on, I need to understand 100% of everything, or else I'm not going to do all in the Mcat. And the one thing I love about doing these walkthroughs of the Blueprint Mcat exams, with me trying to answer them, right? I'm playing the student here. I haven't seen this material in literal decades. And I still do relatively well, right? I'm probably not scoring a 520, but I'm probably close to a 500 with just some basic deductive reasoning.
Right. Yeah, no, that's awesome. And I think it's a really good point too, because here's the thing about the Mcat. I'm sure you tell people this all the time, but the Mcat writers, they know what we know, right? They know what is in the bandwidth of test takers to know and understand.
They tell us what to know, right? It's not like it's a secret.
Yeah, they literally give us a list. And so if something isn't on that list, you don't need to spend a ton of time thinking about whether or not something's going to be tested on the Mcat or not. And if it shows up, you can be like, all right, I'm going to have to rely strictly on what the passage says, but not really go far beyond that. So for that acylation reaction, you can know, all right, if I don't 100% know what that is, it's fine. I just know that it converts an amide to an ester, and that's it.
That's all. That's all you got to know. All right, so then we get to this lovely first question here, which I'll jump into. As the nucleophilic site is serine, at which nucleophilic site is serine most likely to be acylated by penicillin? So we have answer choice A, side chain OH, carbonyl carbon, carbonyl OH, or alpha carbon. So here's where I really struggle as someone who, again, hasn't had this material in forever. Are these super specific, especially stupid amino acid questions? Okay, you obviously need to know, what's my assumption here, right? Again, just using some deduction, is to answer this question, I need to know what the structure of serine looks like, right? What side chain is hanging off of it, and what is going to be attractive to that side chain potentially to be able to answer this question. And of course, again, it's been a while since I've done this stuff, so I don't remember. But that's how I would potentially try to answer it if I remembered that. Am I on the right track there?
Yeah, I think so. And I think I want to also draw your attention to figure 2, to see maybe if that'll give you a little bit more information. Because again, if you don't know something, if you don't remember something in particular, the figures or something in the passage may help you. So I would sort of push you in that direction.
Yeah. So this is the structure of penicillin, right? So it's like, okay, at which nucleophilic site is serine most likely to be acylated by penicillin? So again, trying to look at this and go, okay, what's hanging off here that wants to like give away some stuff or whatever? And I don't know. I don't know.
Yeah, so I mean, I would actually draw your attention to figure 2, because figure 2 shows you the molecular structure of the amino acid serine.
There's serine.
So, yeah, right. And so the thing is, what's important to know for MCAT test takers, people who are looking at this material right now, you can't rely on the MCAT giving you the structure of amino acid serine, because that's relevant information that you probably need to know for test day. But because it's been given to you, you have a little bit more help from the passage in terms of figuring out where on serine do you think there's going to be acylation happening.
Yeah. Yeah. Okay. So is this pretty straightforward answer? How do we get to the answer here?
Yeah. So one of the big I like to do whenever I look at questions is translate it into an actionable task. So for me, this question turns into, all right, which of these different components is going to be nucleophilic? Right. And so nucleophiles are defined as electron-rich species. So we just need to pick an answer that best reflects the part of serine that is electron-rich. And so in that light, we can actually eliminate B and D, because carbons aren't really electron-rich, so they can't be our nucleophiles. And so the difference between A and C, because again, they're both talking about OHs, right? So you need to distinguish the key difference between A and C. And the difference there is that C has a carbonyl near it, whereas A is just by itself. And to determine which one is the answer, you can, instead of trying to think about the reaction that would happen, you could think about what nucleophiles do in general, right? Do you remember anything about what nucleophiles do?
No, I do not. No.
Okay.
I could quickly Google it, but no. I can't do that on the real test, so I won't do it here.
The big rule to know about nucleophiles is that nucleophiles attack electrophiles, right? And so for something to be better at attacking and to order to be a better nucleophile, it's probably going to have to be something that isn't very hindered, right? Something that can fit in a lot of spaces and do that attacking without much like stuff in the way.
Yep.
So between A and C, A is going to be our answer because that carbonyl is going to get in the way. It's likely to stay as our answer.
Yeah, and just looking, right, again, just looking at the structure, you go like, hey, this thing's just dangling out here. This OH is just dangling out here. It has more freedom to stick its head in wherever it wants to go. So I like that.
Yeah, exactly.
All right. So answer choice A, piece of cake. That's what I would have guessed.
Yeah, amazing.
All right, go ahead and read 15 and I'll try to answer it.
All right. Question 15. Early research into the mechanism of antibiotics theorized that it was implausible that the amide in penicillin would convert to an ester. Which of the following explains this doubt? Answer choice A, amides possess better leaving groups than esters. B, esters possesses poorer leaving groups than amides. C, amides are less reactive than esters. Or D, serine contains no good nucleophiles.
Okay. So, I think answer choice. Let me look at 14. Potentially, like the whole nucleophile thing is what we just talked about in the last question. So I may throw out answer choice D, maybe just because it's like, well, maybe we were talking about nucleophilic sites in serine last question. So I'm gonna throw out D just because it sounds too familiar to the last question.
Really.
Early research, so that amide in penicillin would convert to an ester, right? So just knowing biology and chemistry, like things like to stay kind of stable and not take a lot of energy to convert if things are going to be wacko. So if I look at less reactive, amides are less reactive than esters. That maybe sounds true, but is that the answer? I don't know. If something's less reactive than an ester, like, does it convert to an ester and then just converts right back to an amide if that's what it's saying? I don't know. Again, just trying to think through this. Answer choices A and B are basically the same answer, right?
Yeah.
Amides possess better, esters possess poorer. I mean, that's the same answer. So I'm going to leave A and B out of it altogether and just go with C.
Yes, and you are, first of all, totally right on the money there. And I think what you exemplified there is really cool in that even if you don't know the right reason as to why C is correct, if you know explicitly why A, B, and D are wrong, that's enough to get you to where you need to go, right?
Yeah.
The MCAT doesn't really require you to write in a written explanation. So you don't need to always have a direct reason as to why an answer choice is correct. Because I think one of the things I like to tell my students all the time is that there's two ways to get to an answer. You can get the right answer, or you can eliminate all of the wrong answers, right? And so which one you decide to lean on more is dependent on what you know and how you can get to that question. So you're totally right on C. And for those of you who are curious, answer choice C is also correct because of the idea of reactivities, right? In terms of our carboxylic acid derivatives, the big thing to know about them is their relative reactivity. So you can either memorize the order, which is acyl halides, anhydrides, then esters, then amides, and know that you're going to go from more reactive to less reactive and not the other way around. Or you can think about the leaving groups that are there and know that you're not going to move from a bad leaving group to a good one, right? So amides has one of the worst leaving groups, while acyl halides has one of the best leaving groups. And so that sort of explains that memorization of reactivity that you would need to do for that one.
All right. So I got it right.
You did. You killed it.
Don't know anything about it. But again, that's just showing the power of just not freaking out. And obviously it's easier because we're not timed here, but being able to kind of utilize just deduction, right? Sherlock Holmes deduction going through this. All right, question 16. Which of the following must be true if the penicillin-serine reaction, quote, locks in the ester formation as described in the passage? A, the thermodynamic cost of breaking the penicillin-serine ester is larger than the thermodynamic release of amide formation. B, the thermodynamic release of forming an amide is equal to the thermodynamic cost of forming an ester. C, the thermodynamic cost of breaking the penicillin-serine ester is smaller than the thermodynamic release of amide formation. And D, the thermodynamic release of forming the penicillin cyclic amide is larger than the thermodynamic release of breaking the penicillin-serine ester. All right, so long answer choices, which again freaks students out. I'm like, I have to reread those. I have to clarify. I have to figure out what's going on. So this actually goes towards a little bit of what I was talking about with the last question in terms of energy related to doing things, right? Of that this previous question of if it's implausible, are they saying, ooh, maybe there's just too much energy release or not enough or whatever to get there? And so this question is jumping forward to potentially starting to think about that. So how these work. So just trying to figure out how they work. So penicillin, acylate serine structures or residues on enzymes. The amide on penicillin is converted to an ester. All right, so the amide is there. It converts to an ester. So just reiterating that in my head. So the cost of breaking the penicillin serine ester is larger than the release of the amide formation. So this, I think, sounds true because, wait, no, that's opposite, I think, of what I want. It's larger because then it would be easier potentially to go back. Maybe I think that's what I'm reading. All right, so the thermodynamic release of forming an amide is equal. So things are equal, then they bounce back and forth potentially. So I don't like that answer. So I'm going to strike that one out. The thermodynamic cost of breaking the ester is smaller than the release of the amide formation, which is breaking the esters. So these ones are opposite. So I like A or C, potentially the thermodynamic reason forming penicillin. So this one's completely off in terms of just the language and everything that it's talking about. So without even looking at D, I'm going to cross it off because A and C are exactly opposite. So I think it may be one of these. So now I'm hopefully down to 50-50. And again, if the energy to create is smaller than the energy to go back, then to me that signals locked. So the cost of breaking the penicillin serine ester is smaller, so we don't like that one. So I like A.
Yes, that was exactly right. That was totally it. That was beautiful. I couldn't have explained it better myself. It's basic econ, right? Like
Yeah.
minimize cost, maximize gain. And so I love the fact that you were able to use that for that question because that's really the example there. And I think the other rule of if you're stuck between two answer choices and you really like the big thing to do is look at the difference between the two of them, right? And if you have no idea where to go to the things that are the opposite. It's not going to be something that I encourage every single time because there are situations where there are opposites, but those opposites aren't related. But if you have no clue, like if you go to a question, you're like, oh my goodness, I don't know what's going on here. Stick to the two that are the opposite and then go from there.
Yeah.
You killed it.
Same.
That's great.
2 out of 3.
Oh,
all right. 17, go ahead.
All right. Question 17. Which functional group is not found on penicillin? A, amide, B, amine, C, carboxylic acid, and D, thioether. I feel bad because your question that you had to read was so much longer than mine.
That is okay. So here's where again this goes to you know it or you don't know it for the most part. And so again, because I'm so far removed from these types of definitions, I'm like, I don't know. All right, we've been talking about amide groups and penicillin, I think potentially, or were we talking about, so it says specifically, right, there's amide on penicillin. So answer choice A is out. All right, so I'm assuming that's out. So now hopefully I'm down to 3 potential choices. I have no idea what that means. Carboxylic group, I think is something over here, what that looks like. So I'm going to cross out C. And then thioether. I always, like with sulfurs and nitrogens, like there's always funky names for it. But I don't, like thioether, I forget. I'm going to choose D just because I don't like that word, and I hope that's right.
You were so close.
So darn it.
The thioether is that part of the compound that has the sulfur in it.
Yeah.
If you think about it, like an ether is just an oxygen that's between 2 carbons. So the sulfur is the thioether because it's the same thing, but with sulfur instead of oxygen.
You know what? So here's where I ran into the same problem I ran into with the first question. I was looking at figure 2 and not figure 1. If I would have seen that sulfur group in figure 1, then I would have gone with the other way. So
no way. Okay, so that's a really good learning opportunity, though. Like being able to recognize where in the passage you have to go is going to be critically important. So I think in general, when you answer questions, the best things that I like to do is translate the question if it's confusing, figure out where in the passage I need to go. And being able to figure out where in the passage you need to go is very reliant on your ability to have an effective read the first time. And that effective read doesn't mean you understand everything and anything that's included in a passage. It's that you understand what key hot topics are showing up and where you can go in the passage if certain things are being asked about, right?
Mm-hmm.
So being able to map for yourself, like, all right, figure 1 is penicillin, figure 2 is serine. That's enough to help you differentiate and potentially prevent you from making that mistake that you did.
Yeah. Just taking that extra second there.
Yeah, absolutely. But honestly, you did a pretty good job at eliminating A and C because you're totally right. Even given the context of the passage, it doesn't make sense for an amide to not be present.
Yep.
And carboxylic acids literally showed up. Amino acids, like that's one of the biggest structures within an amino acid. So it's good to eliminate that as well. An imine is like this weird double bond with a nitrogen. So for anyone who's listening, that's what an imine is. It isn't super high yield in terms of things to know, but be vaguely familiar with it so that you can recognize it if it shows up as a wrong answer choice.
Okay. All right. And last question here. On the penicillin heteroatom ring, what is the absolute configuration of the carbon labeled 1? All right. So penicillin, figure 1, not figure 2. The penicillin heteroatom ring, the carbon labeled 1 is this one that's also attached to nitrogen and has this whatever group hanging off the side, the COOH here. So the absolute configuration, plus, minus, S, or R. And I don't remember what configuration, the absolute configuration. I don't remember what that refers to.
Yeah, so I think the absolute configuration is, do you remember like the RS configuration? Is that familiar to you?
No.
So the absolute configuration of a compound is more so referring to the orientation of different substituents in space, right? So whether it's like, if I have a carbon with 4 things attached to it, where are those things in relationship to each other?
Yep.
And it's called absolute because no matter how you rotate the specific compound in question, like that's not going to change, right? Because the relationships with each other, like that, you can't change that unless you're literally breaking a bond.
Yes.
So that's the key thing there. One thing I will say is, because I mentioned R and S, right? Which 2 answer choices do you think we can eliminate based on the fact that absolute configuration can only be R or S?
A and B.
Yeah, absolutely. So you can eliminate that, right? A and B, that plus or minus is more so referring to optical activity, and you can only really measure that in a lab. So like not really relevant for us here. So even if you don't have the time to go through each of the steps that you need to do to figure out the absolute configuration, you can still like flag it, pick a letter between C and D, and come back to it if you have time.
Yeah.
Because I'll tell you right now, Ryan, the steps to figuring this out can be very time intensive. For me personally, orgo was not my strong point. So I always took my sweet time when I had questions like this.
Yeah.
So I would always flag them and come back to it.
Yeah, it's funny. Orgo was one of my favorite classes. I even took an accelerated bioorganic chemistry. It was 2 semesters instead of 3.
Wow.
Orgo 1, Orgo 2 biochem. I took it all in 2 semesters because the professor was amazing, and there was just something like that clicked, right? Of just seeing. I'm very visual. I can like see the structures. Even I love talking about. I wish I want to relearn them. Like NMR spectroscopy, like I could do those in like 10 seconds. There was just something about seeing the spikes going, oh, I know what that is. Like just drawing it out. I don't know why. That was like, I was a savant with that. I struggled with a lot of other things, but anyway, but I don't remember SNR.
That's crazy though. Honestly, you're like a unicorn in terms of the pre-med community, because I know a lot more people who are, not a huge fan and have struggles with the visualization piece. And
yeah.
So there's 2 ways you can really do this question, which is you can visualize what's going on and reorient it in space, or you can do what I like to do, which is literally just go by what is drawn and use that to determine what the answer is.
Yeah.
So if I were to teach it to you, Ryan, I'd probably explain the whole like orienting it in space. But for everyone else, for the majority of people who want to do this type of question, there are a couple of steps that you need to do to understand the chiral designation, right? So the first thing you need to do is identify the 4 substituents that are bonded to that carbon. So in this case, we have a nitrogen, we have a carboxylic acid, we have a carbon that's bonded to a sulfur, and a carbon that's bonded to 2 other carbons. And then we also lastly have our hydrogen, right? Remember, the hydrogens haven't been drawn in, but in orgo, you don't have to draw in a hydrogen bond with a carbon, right? The only reason why people don't do that normally is because if you do, then the drawings will just become crazy because it'll have too much stuff involved. So remember that if a hydrogen hasn't been drawn in, that doesn't necessarily mean that it's not there, right? So in this case, those are 4 substituents. The second step is to identify the priorities to those substituents and assign them. And the assignment goes in relationship to atomic number. So the higher the atomic number, the higher the priority. So Ryan, if you don't mind me asking you this question, totally okay if you don't know the answer. Based on the 4 substituents that we have here, do you know which one is going to be the highest or the lowest priority?
Nitrogen's very special, so I'm going to say that's the highest priority.
Mm-hmm. It is. You're totally right. So
which one of these is not like the other?
Yeah, exactly. So nitrogen is going to be our highest priority because of its atomic number, and hydrogen is going to be our lowest because of its atomic number. Now, it gets a little tricky in situations where you have 2 carbons because you actually, at that point, need to go 1 bond away and compare to what's going on there.
Yep.
So with one of our carbons, it's bonded to a sulfur. Well, our other carbon is only bonded to 2 other carbons, right? So we need to look at the one that is bonded to the sulfur as our like carbon of choice, right? That's going to be the one that we need to look at. Okay. So that's going to be number 2. And then the carbon on our dash, right, is going to be priority group number 3. So that's going to be the one bonded to our different oxygens there.
Yep.
Okay. So in terms of our order, we have 1, 2, and 3. And from there, you just need to count from 1 to 2 to 3 and determine the direction you move in, whether it's clockwise or counterclockwise. So you can imagine yourself drawing like a little arrow or moving your finger along that direction.
Yeah.
So
I'm seeing clockwise.
Yes, exactly. But here's the killer. We're not done. And the main reason why we're not done is because our hydrogen, since it's not on a dash, right? We see our carboxylic acid group, that's what's on our dash, that's what's behind us. Our hydrogen is actually facing towards us. So because of that, we need to reverse the configuration that we've assigned.
No.
So, I know, killer. So normally, clockwise is R, right? The mnemonic for it is clockwise is the right way to go for a clock. So if you're going clockwise, it's the R configuration. But because we flipped it, because the Mcat's mean, I guess, it's going to be S, and that'll be our answer.
All right.
Is that so time intensive?
That is way too time intensive. And so I would have chosen C and moved on, and I would have gotten right. The trusty just choose C and move on.
Yeah, these types of questions are really hard to do because they take so much time. And unless you're a savant with visualizing it, you're going to have to go through each of those steps. So if it were me on test day, I would have eliminated A and B, picked a letter, flagged it, and then come back to it if I had time.
Yeah. Ooh, all right. So again, I hope that going through these, right, a couple of them I just read and looked and deduced and guessed sometimes, but a lot of the guessing was gone down to like removing 50% of the answer choices. And so just again reiterating, the MCAT doesn't have to be as scary as you're making it out to be. Just read, learn how to kind of deduce and get wrong answers out of the way to hopefully help increase your chances of picking the right ones.